back to the New Creation channel. My name is Peter, and today I'm joined by Jake Ramgren. You might have seen Jake around before. He's a regular writer for the New Creation blog. He received his undergraduate degree in zoology, and now he's a biology teacher. He gives creation tours at museums, and he's worked on some various research projects, one of which we're going to be hearing about today. Thanks so much for joining me, Jake.
Thanks for having me, Peter. I'm excited to be part of the channel and share some of the research I've done.
Yeah. So today we're going to be kind of discussing a recent article that you wrote that was published on the blog, and it's called Academic Home Improvement, an Opportunity for Creationists. So can you begin by kind of explaining that title for us? What is academic home improvement? What do you mean by that phrase?
It's basically an analogy that I make in the article where I'm talking about how when you have a big project, right, and I use the example, I think, of fixing a bridge or fixing a porch or something that you might have around the house you got to build. You oftentimes need collaboration, and you need to get people together and kind of have teamwork to complete a project or to build something like that. And I kind of make that comparison to research. And in often cases, scientific research, we kind of think of it as scientists who are kind of on their own and they're doing something in a laboratory and they're writing down their results and publishing their results. But in reality, a lot of the projects that scientists have to do takes a lot of collaboration. So you have the home improvement is kind of my analogy, and I'm talking about academic home improvement. So this is just scientists who are collaborating, they're coming together, they're each sharing their own expertise in order to get a project done. And in this case, that project is something called the All Taxa Biodiversity Inventory, which is a really bold attempt by scientists to catalog and document every single species of life in the Great Smokies National Park in Tennessee. So massive projects have been going on for decades now, and it's probably going to go on for decades more. There's a lot of species there. But that's the project that I'm writing about here. And so the title is kind of reflecting an analogy I'm making on how creation scientists specifically should get involved, but it's something that all creationists and all scientists should be aware of. It's happening because it takes all of us message to get this done.
So can you explain for us a little bit about why the ATBI is focusing on the Great Smoky Mountain National Park? What's special about this park? Why catalog the species present here?
Yeah, so there's plenty of articles out there. You can hop on Google Scholar, look it up. There's people who write about why the project is being done. It's actually the second attempt at it. So there was an attempt a few years prior, and scientists decided that they had found the wrong spot, and they ended up moving to the Smokies because they actually estimated that a vast majority of biodiversity that's going to be in basically the eastern United States, a vast majority of life that you'd see there is also present in this park. So it's this massive woods park. It's the most visited national park in America, I believe. And so basically you could go up to Maine, you can go down to Florida, and most of the species that you're going to see in either of those places you can also find in the park. Not all of them, but it basically is a smaller location where you can look at and find a vast amount of biodiversity you find anywhere else. So it was a great place to start. And so if we actually do catalog and we actually do successfully complete this mission, it's going to be the most beneficial for scientists. It's going to be the most biodiversity, all cataloged in one place that you can find. And there were a few places that kind of could have been options, but this is the one that everyone kind of decided on for a bunch of different reasons. But I guess the top one is the vast amount of biodiversity that's in the park was extremely enticing and too good to let it pass.
So can you tell us a little bit about why it is important in the first place to catalog species? So why does it matter how many species of wasps are living in the Smoky Mountains?
You'll find abundance of reasons. First of all, an easy one is because we just want to understand what's in there. We want to understand which species are declining, which ones maybe are salvageable, we can save. We know there's a lot of species in decline. Again, there's been a lot of problems in the Smoky Mountains recently with just different types of pollution, and tons of people are visiting it. So in order to know which species need to be saved, we need to know what species are there. So a lot of conservation efforts would be a big one. Another is there's so one species actually that we know of in subfamily Cryptonae, the one that I researched well, the subfamily I specialized in, we species that we know of, so not ones that we found in the Smokies, but the lepidopterans that they parasitize, that they kill, are crop pests, and so it's actually pretty common practice to um, get parasitoid wasps or other types of parasites or predators, and use them as crop pests. And you release them onto your field, and they kill your pests. And so it's debated somewhat whether that's a good thing because invasive species, and there's a whole conversation out with that. But there are uses for these wasps that we can use, and so we don't know some of what those could be, but there are so many species to catalog. When we get there, it's likely that we'll find benefits to knowing about a lot of them. We and they come in unexpected places all the time. I use an example when I was doing the research with the class. I presented to the class and I said, "Look, when uh, you know, we find medicinal uses for things in the weirdest places, right? We have medicinal use for like horseshoe crab blood, right? Like who knew we'd go to the bottom of the ocean, find these crab-like creatures, and find out that their blood has medicinal qualities. So we find these things all over the place without anticipating it, and there's so much biodiversity here. So we can figure out which species we can save and protect and preserve, and figure out how to do that. And then we can also find unexpected medicinal uses or any other uses, crop pest management. The list kind of goes on. So those are just a few kind of off the top of my head reasons why knowing what's in the Smokies, knowing what these species are, is a benefit for science.
So, how exactly did you get involved in this project? You can kind of explain that. And then, secondly, what was your role in the project? What did you end up doing?
Yeah, so it's part of an undergraduate class, which was a really cool opportunity. Not all colleges will have a research project for you in an undergraduate class, but it was with the school Union University where I got my degree, and we were under Jeremy Bloschke is a professor. He's a entomologist. He's a parasitologist, and so under in his class, he decided he wanted us to do a research project. It's actually kind of an interesting story on why we ended up choosing this one. He had an original plan where we were going to document some species that were in a local park in Jackson, Tennessee, where the school is. There's a park there that Union actually is describing a new species of wasp still that they discovered a few years ago. So there's new species of wasp there. So Dr. Bloschke was really excited to see if we can find anything more interesting in that park. And the park was actually closed for repairs. We get a lot of storms down there, so it was closed for repairs. It was this perfect opportunity. We went in, we set up kind of all these bowls that were nailed to wood, and the bowls we filled up with really sweet water. We put some in the woods and some in the fields. We were going to compare what kind of species we get in each, and that was our research project for the year. It was going to be just as academic. But one time before I even got a chance to go, we all had to go twice to collect our samples. Before I even got to go once, someone showed up and someone had taken all of the wood things we had nailed there and had made a bonfire with them. And so, which they should not have been in the park in the first place anyway. And so, that was very frustrating. We kind of had to, and that was our plan for the semester for invertebrate zoology, that class. So Dr. Bloschke had to kind of change paths late, and he's actually very good at that. He had to do the same thing when COVID sent us all home. He had to go online, and he did some really fun things with that class, even though we were all far apart. So he's really good at that kind of switching gears and adapting to the situation. And so he needed a new research project for us to do, and he had. I'm guessing he was aware of the ATBI happening and actually managed to contact people over there and got a bunch of wasps that were collected in the Smokies and had them sent over here so we could kind of sort them out and have a research project there. So it was an undergraduate class. It's really Dr. Bloschke going out of his way to make the class memorable and give us research experience. And I'm really glad he did. It was a great project.
So explain a little bit how you identify different species of wasps. You mentioned there's a genetic component. Was there also a morphological component? How are you sorting these different species?
Yeah, so we would sort them by morphology first, and then the genetics would come later. Genetics is really the only way you can identify what species it is. But we can't just send a collection of insects and just have them barcoded. So what we had to do was we basically received a collection, and there were tons of them. And this collection had actually been there since 2000, 2001. They had been collected. So I was sorting wasps that were collected the year I was born. Basically, they'd been there for a while. No one had touched them, and they were from uh, the Ichneumonidae family of wasps, which is a massive, diverse group of parasitoid wasps. And we basically sorted them to something that was called morphospecies, as I talk about in the article. That was they just look different. So look at them under a microscope, and if it looks different from anything we'd seen before, we put a number on it and made it a new morphospecies. We couldn't name them; it was not official by any means. And we turned out when we did receive back the data, we had it wasn't perfect. There had been ones that we had sorted different morphospecies that were actually the same species, but they just looked a little different. Maybe one was male, one was female. Maybe they were just individuals that looked a little different. But we looked at them under microscopes. To the best of our ability, we sorted them into morphospecies. Some of them we just were like, oh, another one of these, and just had a massive collection of them. Some of them we only found one. But basically sorted them into morphospecies the best we could. We had a couple of students who actually made this their senior research project as well. So they're a little more invested in the research project, and they had the tedious task of trying to narrow them down to families. And we only really did that because if we found a family, if one belonged to a family that had never been mentioned in the park before, we already know we have a new park record. So we tried to get them all down to family. We had a couple of students on that. I remember they were exhausted. It was hard work for them, and I'm frankly I'm glad I chose to capture alligators for my senior research project, because diving into looking at you're looking at books that are on the older side. You're looking at websites. You're looking at very slight, um, tiny differences in wasps, things on their wing pattern, just little things, and trying to sort them into families. I think they got the most wrong too, because they're undergraduate students, and they did their best. They worked really hard at it, and they got some of them right, which was really impressive. But we sorted them as best we could to families to try and anticipate which ones might give us the best data, and then those are the ones we end up, I believe, getting 95 of them together and sent them over to be barcoded at the Canadian database, where they barcode them and send back the results to us.
So explain to us what barcoding is. Some of our listeners might not know exactly what that is.
All right, so we we didn't do the barcoding. Like I said, we sent them to a barcoding kind of lab, um, and they would basically take a what was called basically a protein in the wasp, called cytochrome oxidase 1, as the protein that they used. And they would look at basically the DNA in that protein. And if it had there's a database called Bold database. You can find this online, and it already has basically the barcodes of lots and lots and lots of species out there. And so if you get something new, you look at cytochrome oxidase 1, you look at the order of the ATCG in the DNA, and if it matched with another's, over 98% of the DNA were exactly the same, you knew it was the same species. But if it's less than 98%, that's suspect. You might think that there's a decent amount of variation in there, so this might be a different species. So we're sending them to a lab that looks at the protein and decides, is this a new species? Is this the same as another species? It will match it all the way. It'll be as specific as it can be. Some of them that came back said, well, we know it's in this genus. We don't know what species it is. But some of them were exact matches to species that were already in the database. So it's not perfect. It's not complete, but we couldn't have done it without that database, without barcoding them.
So, can you tell us what you found? Did you find a new species? Did you find species that you didn't expect to show up there?
Yeah, so we had 95 specimens we sent in. We found plenty of several new park records, a lot of new park records. And then we did find a few that looked a lot like new species. Now, again, the database isn't complete. We don't have every species that we know of cataloged in the database. But we did have examples of a wasp that matched well with a genus, but not with any of the species we had in that genus. And then you look inside that genus, and we only know of two or three species in that genus. So more than likely, that's a new species. So we're fairly confident we've discovered a few new species. The one that basically my part of it was once we were ready to present our data, I basically took a subfamily um, and specialized in that subfamily. So my subfamily was a group called Cryptonae. And this is a little, this is more detail than I give in the article. Again, my article is more focused broadly on the ATBI and use my research as kind of example. But I use subfamily Cryptonae. They're a wasp subfamily that parasitizes Lepidopteran cocoons and pupae, which is a fancy way of saying that they plant their eggs on top of or inside of butterfly cocoons or moth cocoons, and then their babies. And so it's a subfamily, and from just that one subfamily, we found. I can give a couple examples here. We found one that was a species called Gambris incubator, and this species it was a 100% species match, and it had never been seen in Smokies before, so it's a new park record. And then I guess to give one other example, we had one that was narrowed down to the genus Xylophorus. It was a 94% match to the other species in that genus. So not the same species as this one. Very few species of that genus are known, and this one was a pretty distant, like it wasn't close to the species that was already in that database. So fairly certain it should go in that genus, but we're fairly certain that it's a new species. So yeah, we found a few that look like new species of wasps, which is really exciting, and then a bunch of new. I think we had out of 95, we had a good. I want to say we had 20 that were new park records, something in that ballpark.
So, if you want to check out that whole research, we'll have the paper linked down below in the description.
Yeah, the article is called "Using DNA Barcoding to Identify High Priority Taxa from Great Smoky Mountains National Park." It's from the journal Entomological Society of America. It just got published a few weeks ago. I got it, emailed, and so we're excited about that.
So, what do you think are the main benefits of participating in this particular research project?
Well, as you'll find in the article, again, I make that comparison to the collaboration that's involved. So there are different types of collaboration. I cover all of that in the art. Again, that's the article is. I just gave you a lot more information than what's in the article because my article is focused on that collaboration side. So there's different types of collaboration you can do with this project. There are scientists who, like we did for our research, who are contacting people across the world for one research project. So again, make sure we're not just sitting in our labs doing our own research. We're offering our expertise to scientists across the world. Really, we're sending things back and forth to each other. But there's also what's happening at a faster pace is something that's called a BioBlitz. So what they do is in the Smokies, they get a bunch of people together and they go out and they just collect things. They're just finding things and all at once they are documenting, collecting. And it's not just scientists who are doing that. At that point, you're not really breaching a distance of space. You're breaching a distance of education, experience, or whatnot. So you hear people talk all the time about how you should get involved with research, get involved with science, but most people don't have those science degrees, right? They say they want you to love science, they want you to get into STEM fields, but people don't have that experience. Sometimes they don't have 4 or 5 years and all the money to get the degrees that they have to become scientists. But what this project can do is it'll say it doesn't matter what your credentials are, it doesn't matter how much knowledge you have. If you love being outside and you love nature as much as a scientist does, you can contribute to this project. You can go out there and you can help collect things. You can find, and we talk a lot about wasps here, but plants are being documented as well. So you can collect plants, you can collect insects, whatever it is. You don't have to have a PhD, to collect an insect and say, what is this? We should figure this out and bring it to the scientists and collaborate on something like that. So it's connecting scientists from across the world, and it's connecting scientists to people who just love nature, love science, love animals and plants, and so it's an encouraging message, and it's one of those projects that everyone can get involved in.
So going kind of full circle, why is this a project that creationists specifically need to be involved in?
Yeah, so I kind of was inspired by that. So right after I graduated, we had planned to do a trip to the Great Smoky Mountains with actually not invertebrate zoology, but for an ecology class. So it wouldn't have had anything really to do with that project. But I did this whole project without having ever gone to the Smoky Mountains. And our plan, we ended up not going. There was bad weather. And so after I graduated, kind of as a celebration, I had my dad and brother and I just planned a camping trip, and so I spent a few days in the Smoky Mountains, and that's kind of where this article came out. So I just felt like I wasn't done writing about this. I wasn't done with this thinking about this project, and now is going to be where the project was happening. So I talked to wildlife agent that was there. I got like a newspaper from them, kind of like a newsletter on what's going on in the Smokies that had some information on the ATBI. I actually quote it in the article, I believe, that one that I got. And so I was like, man, and at that time, I'm writing for the New Creation blog, and I thought this is such a cool project that if this can breach the gap between scientists who are living across the world from each other, if this can breach a gap between scientists and lay people and people who just love nature, what is stopping this project from also uniting kind of the secular evolution scientists who are doing this honestly? Like, they're trying to find new data that they can explain with their evolutionary model. They're trying to find species that are related and evolving, and that's what they're looking for. But I don't see why we can't bridge the gap between scientists who are looking to explain things from that evolutionary perspective and scientists who might look at the data coming in from the ATBI and interpret it through a creation model. And you won't see a lot of collaboration between creation scientists who are outspoken about being creationists and evolutionists. But this is a project that we can all, receive those species and do research together. And my professor, Jeremy Bloschke, is a young Earth creationist. And he published that article that everyone's going to be looking at, whether you're an evolutionist or a creationist. You can look at that article and know what research is being done. So it's already, in that one tiny case, it's already happened. We have creation, at least one creationist working on it. And so I thought that was a beautiful way to connect our two circles: the evolution, the mainstream scientists, and the creation scientists. And on top of that, it's also just a ton of data that creationists don't want to kind of be left behind in. If we ignore kind of that whole project, there is a whole bunch of data that they're going to be missing out on. And as someone, again, with a degree in zoology, I'm super interested in seeing that species adapt really quickly, or speciation happening very quickly on contemporary timescales, and seeing things quickly adapt into niches that they find. That's something that creationists explain very well, and I'm very interested in. I'm very excited about when I see that, and I feel and I anticipate that there's a ton of that happening in the Great Smoky Mountains, and I want to follow that. I want to see species adapting quickly, because that's stuff that fits right in with the creation model. So it's data creationists should use, but it's also an opportunity to bridge that gap between creationists and evolutionists.
More generally, do you think it's important that creationists are involved in data collection? Because a lot of creationists have kind of this approach that they can kind of sit back and just take the data that the evolutionists have collected and use it themselves. Is this desire to collect data for ourselves fueled by the idea that the data they collect might be biased, or is there something just inherently good about collecting data ourselves that will give us better insight and help advance the creation model?
I'm a huge proponent of both sides of that, both approaches, you could say. I'm I'm regularly reading kind of scientific journals and literature from evolutionists, and I'm constantly kind of reading it from the obviously from the lens of a creationist. So I'm a huge fan of that. I think the positives of that are that you can't be accused of having biased research methods or models because you're using the research collected by secular scientists. So there's a huge and it's also just mainstream science has so much data to use that it's hard to do creation research without paying attention to it. But on the flip side, it's enormously important to be collecting our own data as creationists. And I don't exactly do tons of data collecting myself. I'm one who's doing a lot of reading on my own on a daily basis. You know, I'm a college graduate. I'm a teacher. I'm not a a professor or a scientist going out and collecting things on a regular basis. But I think it's a huge step because you creationists can make great discoveries. The film is Genesis history definitely shows that you know our blog is kind of associated with, or at least we're associated with those scientists. And you can see when we do our own looking, when we do our own seeking, it also kind of validates that we're not just piggybacking, we're not just waiting for the evolutionists to do all the hard work and then we twist it to fit our own models. That's not what creationists are all about. We're actively about going out and finding data that we can then explain without without relying on the science that mainstream scientists are doing. So both approaches to me are very important.
Awesome. Well, thank you so much for joining me. If you're interested in reading the article, you can find that linked down in the description below, as well as some other links to the paper we mentioned earlier and some other material that Jake has produced. Thank you so much for joining me.
Thank you for having me. It's been a pleasure.